proteins. CO2, for the most part, CO2, which gets produced by the metabolic processes of our cells, CO2 is going to be carried as a buffered material in our blood. That's how the majority of CO2 is going to travel. Now, protein structure. Structure determines function. The proteins in our bodies, every single one, has a job to do. Now, enzymes. Listen carefully to what I get ready to say. All enzymes are proteins, but not all proteins are enzymes. Enzymes lower that activation energy needed for a chemical reaction. Their shape will determine their function. Enzymes are going to have an area that is termed an active site. Now, because they have an active site, enzymes are said to be very they are very specific, meaning they only act on a particular type of molecule. For example, the sucrase that I mentioned only works on sucrose. It would not work on lactase, maltose, galactose. It would not work on any other sugar except sucrose. The job, all right, when I have an enzyme, my enzyme can either break a material down or it can build a material up. This example right here, am I breaking something down or building something up?
Building something up.
I'm building something up. There are some molecules, they only fit into this enzyme if they attach to the active site. The enzyme changes its shape. That will activate the enzyme. The enzyme creates a bond between the two molecules. That means we now have a substance that's been created. The enzyme releases it and the substance moves to do what it needs to do. In our bodies, enzymes are not going to be high in number. In other words, if intestinal cells need to make sucrase to break down sucrose, I do not expect that sucrase to be made for every single molecule. It can break down probably 1 million or more sucrose molecules before that enzyme has to be replaced. The reason that enzymes can be used over and over and over is because we can't have them because the body has to do so many things, okay? We can't have them just building up and taking space in the cells, all right? So We keep their number low. They can work at a really high number. When the body needs it, the body will make another one. But other than that, enzymes, hopefully, the goal is that they function like they're supposed to. Now get this. Enzymes are supposed to help a reaction occur quicker, but look, sometimes enzymes need help. The enzymes, what it can do is it can have a little area somewhere on that enzyme where usually a mineral can attach to, and it will work to make that enzyme function better, and that's called a cofactor. If it's going to be something that's organic based, that has carbon in it, we're going to say it's a coenzyme. But anytime you hear the word co means working together. Sometimes the enzymes need a little bit of help. Now, our nucleic acids, the DNA and the RNA, these are going to be nucleotides. DNA, RNA. Nucleotides have that 5-carbon sugar ring, the nitrogen base, and the phosphates. This is DNA, RNA, ATP. So if we look at the phosphate group, we've got the sugar and the nitrogen base. In DNA, the sugar, deoxyribose. In RNA, ribose, nitrogen bases, the ATGC. Adenine, thymine, guanine, cytosine. Which one does that apply to? DNA or RNA? My DNA. Your cell is going to be RNA only. How do they attach together?
G to A, A to T.
DNA does what?
G to C, A to T.
DNA I'm going to have A to T, G to C. That's for my DNA. What do I have in my RNA? A to U, but I still have G to C. What difference does it make? DNA, double-stranded, A to T, G to C. It's either held together with 2 bonds or 3 bonds. What kind of bonds hold it together? Those are going to hold the amino acids together. Hydrogen. Hydrogen bonds hold DNA together. Double-stranded, double helix, A to T, G to C. Does DNA ever leave the nucleus of the cell? No. It's very safe in the nucleus of the cell. But it does need to replicate itself so that the cells can divide. RNA, single-stranded, uses uracil rather than thymine. RNA leaves the nucleus, goes out to those little ribosomes and so forth. Now, this is what I was talking about when we were talking about how strong bonds are. A to T, G to C. What do you notice about the number of bonds between them? How many do you find between A to T? 2. How many do you find between G to C? 1, 2, 3. This is termed a base pair. And a DNA strand can be millions of base pairs long. Now, what is the structure of DNA? The double helix, where it looks like somebody took a ladder, okay, held it in place up here, and then twisted. But that DNA, as it coils, as it twists, is not uniform because of this bonding. Because one has 2, one has 3. That's the reason that it takes the double helix. These bonds have to be broken for this DNA to either replicate itself or code for a protein. What do I mean by that? Let's go back to my insulin and the pancreatic cells. In the nucleus of every cell, it contains DNA. How many molecules of DNA are found in the nucleus of every one of those cells?
Very good.
46 molecules of DNA, because you have 46 chromosomes. Is the gene for the insulin found on every one? No. It will be found on one of those chromosomes. Note my terminology. What's the remainder of them doing? They're dawdling. Okay? Because does my pancreatic cell need to be coding for anything else? No. The only thing I need it to do is focus on the products. It needs, insulin's not the only one, but y'all just hang with that, okay, for a while. So here's what's going to happen. In the nucleus of that cell, that strand of DNA, it's going to have a little gene, a section, like if it's this long, okay, there's a section on it that codes for that protein. That section will remain open, and the RNA comes in, reads it, codes, goes out, makes the insulin, read, code, go out, make the insulin. Are you following what I'm saying? So, something somehow has to break those bonds. Otherwise, that DNA, all 46 of them, inside that nucleus, they would all be coding for stuff. We can't have that. Okay? So those bonds are extremely strong. When the DNA has to unzip so that it can replicate itself, it does. But other than that, those bonds are strong. Now the RNA, single-stranded, we've got uracil, and then the job. Interpret the DNA. Go make the proteins. This is going to give us the primary structure. What is the primary structure?